Welcome everyone to this year's NVMW presentation from our side. So we are going to talk about the capacity of DNA-based data storage under substitution errors, and this is joint work with Paul Siegel from UCSD, Antonia Wartacé from Technical University of Munich, and Eitan Jacobi from Technion. So to understand first our work, we want to first look at the DNA storage system and how they typically look like. So say if we want to store some binary data, which is given in form of bits, and we want to store it in a DNA storage system, the first step is to encode it via an appropriate encoding mechanism to multiple DNA strands. These strands are then synthesized, which means that they are artificially created and physically stored in a storage container until we want to retrieve the data, which means we want to read it. And this is done by sequencing the data, which means that we take the DNA strands that we have stored in our container and read them to DNA reads. And after what we have read, we want to use this data to decode the original user binary data. So there are several aspects that are interesting in the system here. First of all, that is, as you have seen here, we store the data on many short strands, in this case 3, because the synthesis technologies current can only work with sequences of length maybe 100 to 1,000, and same applies to sequencing technologies. Second of all, you see that there are errors within this sequencing process, which are highlighted in red here, which means that we need to have an appropriate error correction mechanism in order to correct the data successfully. And finally, interesting enough here, we may read some sequences multiple times and also some of them none times, which we will see later. So what is the mathematical channel model behind this system? So say, as in our case, we have 3 sequences which are stored, and we are now sequencing the data. What we obtain is that every output sequence is a random draw of an input sequences plus some error, which are highlighted in red here again. So in the first read, I drew the second sequence, then I drew the third sequence plus some errors, then I drew again the third sequence. Luckily, I was drawing the first sequence again, and in the last read, I draw the third sequence. And as we see here, it may happen that we draw some of the sequences none of the times. Notice that these colors here indicate the origin of the sequences. However, they are not known to the receiver. All right, so mathematically, we can say that every output sequence is an input sequence with a random draw plus some error vector, where this plus should be understood as the addition over a circular ring. All right. Now the error vectors, they have error probability p, and in this work, of course, the alphabet size is 4 because DNA consists of 4 bases: A, C, G, and T. All right, so what is the channel input? We have M sequences, in this case 3, each of them having length L. And if we stack all of them together, we obtain the matrix X. And we have a parameter beta, which controls the density of the system. On the other hand, we have the channel output, N sequences, each of length L. So here we have 5 sequences, and we can store them to the overall output matrix Y. And we have another parameter C, which controls the number of output sequences that we read per input sequences, which would be 5/3 here, because we read every input sequences 5/3 times on average. All right. To look at the communication system, it looks like follows. So say we have a message W, which is the data that we want to store. It first goes through the encoder, then it traverses the channel that we just looked at, then it goes through a decoder, and hopefully in the end, whatever we decode is equal to the same thing as we have stored originally. All right, so a code in this sense is then a matrix over the alphabet 4, which M rows and L columns, because we have as many nucleotides stored. And the code rate is just log the number of code words as usual, divided by the number of channel users, and this is the total number of nucleotides. On the other hand, we have a decoder which takes a matrix of size n times L, which are all the red sequences, and maps them back to a code word. And as usual, the error probability is just the probability that whatever we decode is not equal to what was transmitted. All right, so this brings us to a definition of channel capacity. And we say that, as usual, a rate is achievable if there exists a code of rate R, such that the error probability vanishes as the channel dimensions tend to infinity. And as usual, the capacity of such a system is just the supremum of achievable rate, and therefore marks the limit for which we can transmit reliably on this communication system. All right, what we did in this work is we computed the capacity of this channel, and it looked like follows. So we have a weighted summation here, which is weighted with a +1 distribution with parameter C and realization D. And this is weighted with a term which we call multi-draw capacity, which is just the capacity of a channel which repeats the input sequence D times and then transmitted over a Curie symmetric channel with error probability P. Additionally, we have this rate loss term here, which is according to the randomness that we don't know at the receiver how to assign the output sequences with the input sequences. And if we plot this capacity over different channel parameters, we see that intuitively, as the more sequences we draw, that means the higher C is, the larger the capacity becomes here. All right, this is all for this short summary talk. I hope I raised your interest, and I will be happy to talk about this more detail on the conference. See you.